RNA molecules have many roles within the cell, but the thing is, most of these roles are related to protein synthesis. And I just wanted to take a few minutes to do a quick review of the many diverse ways in which RNA takes part of our cell's life. First, let's talk about the types. You have the three main types: the messenger RNA, transfer RNA, and ribosomal RNA. And we've already talked extensively about what they do within protein synthesis. Messenger RNA carrying the message from the nucleus into the cytoplasm, transfer RNA decoding the message and attaching themselves to the amino acids and bringing them to the ribosome where they're actually going to do their job. And you have the ribosomal RNA, which is part of the ribosomal units which are going to be doing the actual catalysis of protein synthesis. And all of these molecules are coded by DNA and specific DNA polymerases, like we talked about in the previous lecture series. But you also have other types of RNA. We also mentioned some of them throughout the protein synthesis lecture. You have the small nuclear RNA, which has a structural and catalytic role within SNRPs in the spliceosomes, which are involved in RNA processing. So it's an RNA, again, to process the messenger RNA. You also have SRP RNA, which are part of the proteins which act like the molecules which recognize the signal particles or the signal sequences of the amino acids of the early protein as a message to drag the protein and the ribosome to the wall of the Roth-ER so that the protein can be made for exportation from the cell. You also have several types of RNA which are involved with gene expression control, including siRNA, miRNA, and piRNA. And we'll talk about this in more detail within the gene expression lecture series. But it's actually very phenomenal that all of these kinds of RNA have evolved. And you also have viral RNA, or RNA that's found inside virus. Then you also have ribozymes, which are RNA molecules which are involved in acting like enzymes throughout the body. And finally, you have viroids, which are things that act kind of like virus, but are just basically naked RNA that's self-replicating. All of these RNA types indicate something interesting about the molecule. It can do many different jobs because it can assume many different structures. And for example, then you have RNA acting as an information carrier or genetic material. You have RNA having structural roles, having catalytic roles, acting like a director particle, the way proteins sometimes do, a regulator, or as a recognition particle. And so RNA can have multiple jobs in a cell. So now that we've talked about this, it's time to revisit that age-old question: what is more important, DNA, RNA, or protein? Well, you wouldn't have the DNA without the proteins, because proteins are involved in the structure of DNA and in the replication of DNA. Proteins are also involved in transcription because they determine which parts of the RNA are going to be available for transcription. Remember the transcriptive factors? And you also have proteins involved in the process of creating RNA, like RNA polymerase, for example, and all of the other enzymes and transcription factors, elongation factors, and everything that we talked about. And you also have proteins in the process of protein synthesis itself. And so it looks like proteins are very important. But you also have DNA, which is the message that is necessary to make all proteins and all RNA in the body. And so without the master plan, you wouldn't have proteins or RNA. Now, what about RNA itself? You need RNA to make proteins because it's the bridge between the DNA and proteins. But you also need the RNA to make DNA sometimes. For example, viruses, which are RNA viruses, start off the RNA, but actually to make the actual DNA. But remember that without the RNA, we wouldn't have the proteins, which then are part of the structure of DNA. And so it's like all of them depend on each other. The whole central dogma idea where DNA stores genes which make proteins, and that happens through the RNA bridge, only makes sense within the context that they all depend on each other. But if you had to pick one of these molecules as the original molecule or the most important molecule, which one would you pick? Would you pick the one which is what you're made of? Everything, 90% of the stuff that your body does is made of proteins. Would you pick the one that includes the master plan to making the proteins or the ribosomes or the RNA? Or would you pick the RNA, which acts as the bridge and can have so many multiple jobs? Now, when life first evolved, you have the chicken and the egg thing. Could you have DNA without proteins? Could you have proteins without DNA? Could you have RNA without either of them? That is the question that you really have to ask yourself. To answer this age-old chicken or the egg question, we actually have to look to the early life on Earth and to try to answer the question. And we will visit this when we do this history of life later in the year. But for now, just think about the complexity of these molecules. How could a protein, which is so complex, develop by itself? The chances are very, very small. Just look at the probability. If you have a simple protein with 37 amino acids, and they're strung and next to each other, for every amino acid, there's a 1 in 20th chance of that being the correct amino acid in the sequence. But since there's 37 amino acids, the total probability of that event is 1 divided by 20 raised to the 37th power, since each event is independent from each other. If you actually put that in the calculator, you will see that that number has more numbers than there are seconds in the history of the universe. And that means that's a very abysmally small chance of this actually happening by chance, even if you have millions of trials per second. And so it makes it very, very hard to believe that a protein would develop by itself. And even if it did, it wouldn't necessarily explain how would you get from that to a DNA. You can't actually go backwards that way. You can't get a protein and make DNA out of that. It makes more sense to make proteins out of genetic material. But then which one of the two would actually develop first, RNA or DNA? Would it be a complex double helix molecule that scores upon itself and needs proteins most of the time for structural support and replication? Or would it be a simpler, one gene, single-stranded nucleic acid molecule that can act in multiple roles, including catalytic, structural, and genetic material transference, or messenger roles? Clearly, you start seeing that RNA was a simpler molecule that could have evolved by itself and first. Think about the fact that the energy molecule of the cell, ATP, is the same molecule that makes the nucleotides for RNA. There seems to be a recycling here of early molecules. Then think about the fact that RNA has multiple jobs. Think about viruses, which are RNA based, and they can do most of the things that the viruses do just because they have a single genetic material molecule called RNA. And think about viroids, which are basically just RNA, self-replicating RNA, and they have properties which mock properties of life. Well, I wouldn't call that actually a life, or I wouldn't necessarily call the first life form of one single self-replicating RNA surrounded by a lipid membrane as a life form, but it is something that makes it more likely to make copies of itself. The membrane will protect the RNA, and the RNA with self-replicating properties will actually make more copies of itself. So molecules like that would have an advantage over other molecules in the early formation of the Earth, and that with itself would make evolution take over. And we will talk about that when we do the evolution of life later in the year. But because RNA has so many diversified roles, and because it has a simpler structure than DNA, you can start to think about RNA as the original molecule of life. And think about it. You could get one RNA which contains one gene, and then double that to make a double helix. And now you have something that looks like DNA by base pairing rules. And what if you get that and you connect with another piece of RNA that underwent the same process for a different gene? And what if you do that and connect that to another piece of RNA that did the same process with a different gene? Before you know it, you're going to get something that looks like the prokaryotic chromosome, which are just basically a string of genes next to each other. And so you can see how DNA could have evolved from RNA. And then once you have DNA, it's easy to understand how the protein synthesis process will actually take place, and how RNA can actually help catalyze the production of proteins. Now, this didn't evolve from thin air. Complex mechanisms like that didn't evolve all at once. It was small steps that made this protein synthesis actually evolve. And we'll talk about things like that when we do the history of life later in the year. But the key thing about this that I wanted to go over is that clearly RNA may have been the first original molecule of life because it's simpler than the other two, and because you can see how proteins can come from RNA and how DNA can come from RNA, but it's hard to see the opposite happening, or the DNA could explain how RNA came about, but you wouldn't start with something more complex. You would probably start with something simpler to begin with. I also wanted to point out that mutations can happen throughout this process, which is what drives this whole evolution in the first place. Especially early on when mutations were running rampant, because we didn't yet have the mechanisms to protect us from it. Look at prokaryotes and how fast they mutate, because they have fast transcriptions, fast translations, fast DNA synthesis. A lot of mutations will gather very fast because they don't have the mechanisms to protect the cell from mutations that eukaryotes developed later on. Mutations can happen during the DNA copy process, where mistakes happen on the actual replication. Mutations can happen during transcription, where something is incorrectly transcribed. They can also happen during protein synthesis. Maybe the wrong amino acid is put in place. Lots of things can cause a protein to end up not being the way it's supposed to be. Incorrect folding on post-translational things. Incorrect sniping or putting together of subunits. Lots of different things can cause the protein to end up as a failure or as a different protein that it's what it's supposed to be. And that allows life to create more variety than you can possibly imagine. And that's why there's so much different types of life forms on Earth. Remember to review mutations. I did a lecture about that within the nucleic acid lecture series. So go back to that on the chromosomal inheritance lecture series. You can go back and revisit the idea of mutations. And one last video, we're going to talk about the history of the research, or the early research that led us to the discovery of these protein synthesis mechanisms. I'll see you guys then.